Good morning, students. Today we'll study about clotting of blood. As you know that if we are having a minor injury or a cut, then after some time the blood flow stops or the bleeding stops. So it stops just because of the coagulation of blood or the clot formation. How does this clot is formed, and what are the components that are involved in the process of clotting? Then this we'll study in today's class. We'll discuss about clotting of blood in our body, in the injured parts of the body, and then in a test tube. If the blood is kept in a test tube, what happens to that blood transfusion, what is blood transfusion process, different types of blood group, who has given the term blood transfusion, and what is the role of blood group in the blood transfusion process. All these things we'll study today in today's class. Then the compatibility factor is that to which groups the blood can be donated to, which groups, or which blood groups are compatible with each other, and non-compatible groups. So this before blood transfusion we have to check the compatibility of the blood group with the donor or with the recipient. Then the RH factor. What is the RH factor? We say that my blood group is A positive or O positive or B positive. So what is this positive or negative factor? What is the role of this factor in the process of blood transfusion? What is the role of this factor in our body? So in today's class we'll discuss about these minute details also. So let's begin with the first or the major topic, which is clotting of blood. This question is very important for examination point of view, as you have to write about the protein, about the enzyme, whichever is in active state, which mineral is required, then what are the vitamin required, role of vitamin in this. So all the terms related to the clotting may be asked in objective type or descriptive questions. So descriptive means the section B part also. So let's start with the process first. Just understand the process of clotting, then it would be easier for you to answer every question. Let's start here. Clotting, whenever there is a cut or wound, from that area an enzyme is released which is termed as thrombokinase. Thrombokinase or thromboplastin. It is the two names are for the same enzyme, thrombokinase and thromboplastin. It is released at the site of the wound or the injury part. Released at the site of injury. Here, what is the role played by this thrombokinase or thromboplastin? This enzyme, it is an enzyme. ACE, if it ends with ACE, so it is an enzyme. This enzyme catalyzes our process and converts prothrombin, which is in inactive stage, from inactive state to active thrombin. This conversion is always in the presence of calcium ions. Calcium ions in our body, the mineral which is responsible for the clotting process is the calcium ion. The prothrombin, inactive prothrombin gets converted to active thrombin in the presence of calcium ions and this enzyme which is thrombokinase. So this conversion, now the thrombin which is formed, it reacts with fibrinogen, and again this process takes place in the presence of calcium ions. So the thrombin when reacts with fibrinogen is a soluble component, and it forms fibrin which is insoluble. Now this two steps are involved in the process of clotting. First, thrombokinase, which catalyzes the reaction of prothrombin to thrombin, that is inactive prothrombin to thrombin in the presence of calcium ions. The catalyst is thrombokinase. Again, the thrombin formed here, it reacts with fibrinogen, which is a soluble component, reacts with fibrinogen in the presence of calcium ion, converts it into fibrin. Fibrin is a network. It forms a network or a mesh like this. The blood cells, they get trapped in these mesh or the networks, which gives a red mass to the wound, or the red appearance to the wound, and that appearance is termed as a clot. Red mass, because the RBC or the blood corpuscles, it get trapped in this network, in this mesh, and the plasma, it oozes out and it is termed as serum. The clear fluid, which is the plasma, it is termed as serum here. It oozes out from this mesh, the blood corpuscles gets trapped, forming a clot, stopping the flow of blood or the bleeding, and the trap is formed. It stops, it traps this network, traps the blood corpuscles and forms a clot. Serum comes out, clear fluid comes out, that is serum. Now here, I have not explained the role of vitamin K. Role of vitamin K is, it helps in the formation of prothrombin. So deficiency of calcium ions, deficiency of vitamin K, it may lead to improper clotting process or delayed clotting of blood, or delayed clotting of blood may lead to excess blood flow, and excess blood flow may lead to death of the person also. So this is all about the clotting. I'll repeat this. Just a second. Yeah. Clotting, it takes place in the presence of an enzyme, thrombokinase, in the presence of of a mineral, which is calcium ion. Prothrombin gets converted to thrombin. Prothrombin is inactive, thrombin is active. These are proteins. One more thing, sometimes if it is asked what is the protein in this, so students, they are not able to write what is the protein in the clot. So fibrinogen and this thrombin, it is they are the proteins. So the thrombin, it along with the fibrinogen, it reacts with the fibrinogen in the presence of calcium ions, form fibrin. Fibrinogen is soluble, fibrin is insoluble, it forms a network, it forms a mesh. In the mesh, the clots and the blood corpuscles, they get trapped, the clear fluid or the plasma, it comes out as serum. So red mass is the clot and the clear fluid is the serum. So this is in some person, the hemophilic person. If a person is suffering from the disease hemophilia, it is due to it is a genetic disorder. It is inherited from parents or grandparents. So the hemophilic, if due to deficiency of some certain vitamins in that, since it is a genetic disorder, it causes deficiency of some vitamins. Which vitamin? Vitamin is vitamin K, and it results in less release of this enzyme, which results in delayed clotting of blood or excessive flow of blood. Even excessive bleeding may lead to death of the person. Again, one more thing in case of viral dengue. Dengue, actually it is not dengue. Here there is tremendous decrease in the blood platelets. decrease in the blood platelets. This is the clotting, the complete process is just because of platelets. So decrease in platelets leads to less blood clotting, leads to excessive blood flow from the injured part, or even death of the person, or excessive blood low blood count, low blood platelet count may lead to death of the person. So this one. Next topic is blood transfusion. Blood transfusion. Blood transfusion. Blood transfusion is a process at the time of need. At the time of need, when there is an accident, when there is bleeding or excessive bleeding in some person, if there is a deep cut in a person's body or excessive loss of blood takes place, then at that time, or in operations, if a person undergoes an operation, or if a person is not able, if the body is not able to form blood, then also the blood is transfused from a donor to the recipient. That transfusion, that transfer of blood from a donor, from a healthy person to a diseased or an operated person, then and that is termed as transfusion. But before blood transfusion, we must take care of few things. What is the blood group? What is the blood group? If what is the positive and negative RH factors? RH is positive or RH negative. That positive and negative factors. Then if the blood group is compatible or not. So before transfusion, these things must be considered. So let us study about the blood group and their compatibility. Carl Landsteiner, he has given the blood group system, and the blood group system is blood group is ABO system, ABO, and according to him there are total 4 blood groups: AB, A, B, O, and one more is AB. Depending, and these blood groups are named based on the antigen present on the blood corpuscles. Antigen, if antigen A is present, then the blood group is A. If antigen B is present, then it is B. If both absent, then the blood group is O. Then what happens if both the antigens are present? Both present, then the blood group is AB. On the basis of presence and absence of blood antigen on the blood corpuscles, these 4 blood groups are formed. If the antigen is A, then blood group is A. Antigen B, blood group is B. And both the antigens are absent, blood group is O. Both are present, blood group is AB. They release but all the blood based on that, they produce antibodies, which is just opposite to the antigen present on their body. So the antibody present here, for A the antibody is B, for B antibody is A, for O it is A and B both, for AB it is antibody is O. So this is the blood group, or various all 4 types of blood group given by the scientist Carl Landsteiner. And now the term is compatibility. Compatibility, which blood group would act as a donor, which would act as a recipient, universal donor, universal recipient. So let's write the compatibility. Next term is compatibility, donor or recipient groups. All the 4 blood groups I'm writing here. For the recipients also. Now this one. The A blood group, it could donate to blood group A and not to B, not to AB, not to O. Blood group B, it would donate, to AB also it could donate. This B can donate to blood group B and AB, not to O. AB, it cannot donate blood to A, not to B, not to A. It can donate blood only to AB. O, it can donate to A, to B, AB and O, all 4. So since it can donate blood to all 4 and it is termed as universal donor. Universal donor O. AB, it receives blood, you can see it receives blood from all 4. So it is universal recipient. AB is a universal recipient. Again, I am repeating this complete process. Compatibility, which blood group is compatible with which one? All 4 groups as donor, all 4 groups as recipient. A can donate blood to A and AB both, not to B, not to O. B can donate blood to B and AB, not to A and O. AB can donate blood to only AB, not to rest 3. O can donate blood to everyone. Since it can donate blood to all 4 the blood groups, so it is termed as universal donor. AB receives blood from all 3, so it is universal recipient. And this A and B blood groups are based on the antigen present on the surface of the blood corpuscles. So this is, then the last topic is RH factor. We use the term A positive, O positive, or this A negative or O negative. This negative, this is not a cation or anion. In bio it is positive and negative. We can write A positive or B positive also, like this also we can write. So this positive is this factor, RH factor, which is present in rhesus monkey. It was first discovered from the rhesus monkey. And this factor, if this factor, this antigen, it contains D antigen, antigen D, or RH factor is also termed. So antigen D or RH factor, if it is present, then the person is positive for that blood group. If that antigen is absent, then the person is negative for that blood group. So this is about the positive and negative. Then one thing I have skipped, this one, thrombokinase or this thromboplastin. It is also termed as Stewart factor. Sometimes they ask what is, so thrombokinase or thromboplastin, it is termed as Stewart factor. For 1 marks question it may be asked. So this is all about the clotting of blood. Clotting of blood in a test tube. We have forgot this. We have skipped this one. In the test, if the blood is kept in a test tube for some time, if the blood is kept in a test tube, then what happens to the blood is the clot is formed like this. the entangled mass of the corpuscles is formed, and the serum, it collects as a clear fluid. Above this clot, the serum is collected, and this is a clot. This is in case of test tube. If the blood, this is blood. If the blood is kept in a test tube for some time, after some time we will observe that a clot is formed, a red mass is formed, which is a clot, and the serum collects as a clear fluid. Thank you students for attending class.